What is life? Take a moment. What comes to your mind when you think about it? Is it the way plants grow? The way your body heals a wound? Or perhaps the DNA that stores everything about who you are? No matter how different we all seem, whether insects, birds, plants, or humans, there's one thing that connects us. It's DNA, the blueprint of life. Not only humans, but also the tiny creatures that surround us share DNA as a fundamental component of life by carrying instructions that tell cells what to do and how to do it. But here's the real question: how is this DNA copied, protected, and passed on generation after generation? Everything mentioned here is linked with one crucial enzyme, that is polymerase. It catalyzes the synthesis of DNA or RNA by assembling nucleotides. Whether replicating DNA or transcribing it into RNA, polymerases drive these essential processes. There are different types of DNA polymerases in prokaryotes and eukaryotes as shown. Each polymerase has a specialized function that ensures efficient and accurate DNA replication and repair. Let's focus on DNA polymerase 1 in prokaryotes. It binds to the primer template junction, a structure with an RNA primer attached to a single-stranded DNA template. Polymerase 1 binds with high specificity, ensuring that the primer aligns perfectly with the DNA template, setting the stage for nucleotide addition. This binding occurs through molecular recognition involving hydrogen bonds, van der Waals forces, and electrostatic interactions. DNA polymerase 1 has a structure similar to a right hand with main subdomains: the fingers, palm, thumb, exonucleus, and active site. DNA binding occurs in the cavity formed by these subdomains. The active site is based on conserved amino acid residues of the palm subdomain. Let's zoom into the active site, which has catalytic magnesium ions, which is represented by green spheres here, along with nearby amino acids. The actual nucleotide addition takes place in this region. In the catalytic core, two magnesium ions stabilize the incoming nucleotides by coordinating with their phosphate groups. One ion activates the 3' OH group on the growing DNA strand, while the other aligns the incoming nucleotide for phosphodiester bond formation. Detailed mechanism is shown in the later part. Key amino acids like aspartate and glutamate coordinate the magnesium ions, stabilizing the active site. Positively charged residues such as arginine and lysine interact with the negatively charged DNA backbone, ensuring precise alignment for catalysis. Once aligned, polymerase 1 catalyzes the chemical mechanism of DNA polymerization. An aspartate residue near the incoming DNTP, that is guanine here, acts as a base, abstracting a proton from the 3' OH of the growing DNA strand. This converts the 3' OH into a more reactive nucleophile. The electron-rich 3' oxygen attacks the alpha phosphate of the incoming DNTP, that is adenine here. This creates a trigonal bipyramidal penta-coordinated transition state around the phosphate. The penta-coordinated structure collapses, forming a new phosphodiester bond between the 3' OH of the existing strand and the 5' phosphate of the incoming nucleotide. Coordination with the second divalent metal ion like Mg2+ stabilizes this release, driving the reaction forward. Thus, the energy for this reaction comes from the hydrolysis of the triphosphate group, releasing pyrophosphate, the leaving group. Polymerase 1 has an exonucleus activity that helps maintain accuracy. If an incorrect nucleotide is added, polymerase 1 backtracks, removes the error, and resumes synthesis. This proofreading reduces errors to 1 in a billion nucleotides. Along with DNA polymerase 1, other polymerases too are essential for biotechnology. Beyond their role in replication, these enzymes drive innovation in PCR, DNA sequencing, molecular cloning, and genome editing, proving to be the masters of molecular machinery.